We're constantly urged to consume less sugar for good reason. The extra calories can result in weight gain, and sugar also drives up blood glucose. And that's a risk factor for diabetes. Various sugar substitutes such as aspartame, sucralose, acesulfame, potassium, saccharin, cyclamates, stevia, these are all available, but they have issues of their own. The search is on for a sugar substitute that can replace sugar in terms of sweetness without contributing calories or increasing blood glucose. Allulose is a naturally occurring sugar found in small amounts in wheat, raisins, figs, and maple syrup. It's about 70% as sweet as cane sugar, but is minimally absorbed through the digestive tract and mostly passes through the body unchanged. Since it isn't absorbed, its caloric contribution is essentially zero, and its effect on blood glucose is also minimal. That means it can be a useful sweetener. Furthermore, allulose interferes with enzymes in the small intestine that normally break starch down into glucose for absorption into the bloodstream. That means the undigested starch passes from the small intestine into the colon, where it is digested by bacteria, with the bacterial digestion products mostly passing out of the body. Short-chain fatty acids are an exception. These are absorbed from the colon and actually may have health benefits, such as improving immune function. On the other hand, the bacterial digestion also produces gases that can cause bloating and discomfort. The extraction of allulose from foods is not practical, but an enzyme, tandem D-allulose 3 epimerase, can convert fructose into allulose. This is found in a number of bacterial species, and the gene that codes for this enzyme has been isolated, cloned, and inserted into the genome of another bacterium, Bacillus subtilis. When these multiply, they produce large amounts of the enzyme that can then be used to convert fructose, which is produced from cornstarch, into allulose. Researchers at Metropolitan University in Toronto sought to investigate whether allulose added to instant white rice would reduce the glycemic effect of the rice, a measure of its ability to raise blood glucose. The scientists used an in vitro digestion system, meaning that chemicals in glassware were used to mimic the digestive process. They found that mixing a serving of rice with 40 grams of allulose reduced the amount of glucose that was absorbed by about 37%. Less allulose was much less effective. The problem, however, is that 40 grams of allulose is more than the suggested intake at one time, because this dose can cause gastrointestinal symptoms. While this is an interesting study and does show that the addition of allulose to starchy foods can lower the impact of that food on blood glucose, it doesn't have a great deal of practical significance. In vitro studies are far from clinical studies in humans, and such shortcuts to controlling blood glucose are not likely to have much impact. It is the overall diet that has to be controlled, meaning reduced intake of sugar and starchy foods. For example, just switching from white rice to whole grain brown rice will reduce the amount of glucose that enters the bloodstream. That for today is our Cup of Joe.